Good morning, everyone. So, the theme of my poster was the possible origins of yeast prion domains from polyglutamine and asparagine tracts. And I must repeat my what head of my laboratory, Professor Thurman, on it. I'm not sure that this is really related with origins of life, except in a very narrow sense, which I will sort of touch upon at the end of my presentation. Briefly, amyloids are fibrillar protein aggregates which have a very specific cross-beta structure, and they can grow by addition of monomers, converting them into amyloid form. And the proteins that form amyloids are unrelated with each other. Many proteins can do this, but the amino acid sequences that drive amyloid polymerization can be grouped into two rough classes, and those are either short stretches of hydrophobic residues or longer stretches of polar residues, such as glutamine and asparagine, or these stretches can be not homogeneous but heterogeneous. They have lots of asparagine and glutamine, but they're interspersed with other residues. And what's interesting about amyloids is they are connected with a lot of diseases in mammals, and these amyloids seem to be toxic. And yeast is a very useful model for studying amyloids because lots of proteins able to form amyloids were discovered in yeast, and some of them are prions, which were described in detail in Professor Thurman's lecture. But briefly, prions are amyloids which are able to be fragmented by chaperones, and for this reason they can infect cells without amyloids, and they can be inherited during cell division. And in previous work we discovered that interspersing, amyloids made up of proteins with polyglutamine domains are fragmented inefficiently, while amyloids that have interspersions of other amino acids, and it turns out that nearly half of the possible amino acids do this, they can improve amyloid fragmentation, thus making normal amyloids more like prions. And so because of this, we thought that possibly prion domains, which are usually interspersed glutamine asparagine rich, evolved from pure polyglutamines by accumulation of mutations. And what's interesting is that since the DNA encoding of polyglutamine stretch is homogeneous, mutations can easily be amplified in polyglutamine stretches, thus the level of interspersion can grow. So I do not really have time to talk about the factors which confirm this hypothesis. Basically, there are several lines of evidence that suggest that the ability to form amyloids and also the toxicity of amyloids decreases when mutations accumulate in pure polyglutamine stretches. And also, we did a rough survey of different homologous proteins from different strains of yeast, and we seem to see that the ancestor sequences of many prion and prion-like proteins might have pure polyglutamine ancestral sequences. Why is this important? This is important because prions, as an element of protein inheritance, they're always discussed whether they're a blessing or a disease, or more specifically, was there positive selection for prion properties? And our hypothesis indicates that possibly emergence of prionogenic properties was an inevitable consequence of the existence of numerous proteins with polyglutamine or polyasparagine domains in the proteome. And indeed, proteins like this, in some organisms, they can take up 10% of the proteome, while in others, several percent. So they're very common. And to conclude, possibly the ability to form amyloids is probably an ancient feature of polypeptides, because it is thought that nearly any protein can form an amyloid in a specific set of circumstances, and also short polypeptides are known to form amyloids. So our work touches upon the evolution of this ancient trait of polypeptides, even though in a more modern setting as yeast. So thank you very much.